So for people waking up to the news, what happened today, sir?
Oh, I got a call from Stockholm at, I don't know, 4:30 in the morning, something like that, saying they have good news, and it's and I knew it was going to be a transformative day because the Nobel Prize is huge in the world of science.
Great. So what went through your mind as you get that call and you knew it was the committee?
Well, I just kept repeating in my mind, this changes everything because, the Nobel is just mythic in how it transforms the life of people who are selected.
Congratulations.
Thank you.
So what did you win? What work earned you this recognition?
It was work that we thought of as sort of trying to early genetic analysis of how organisms choreograph their cells to produce complicated organisms like having a brain and an intestine and muscles and how does that get choreographed from the DNA. And so we were working on the genetics of that. And in this case, the beauty of genetics is that you don't know what answers it's going to tell you. You look for variation that looks different than what you normally see. And so from our mutants, we figured out that what was regulating this process in this little nematode was a gene that was much, much, much smaller than any gene that had ever been discovered in any organism. And it was hard to prove, but then comparing different genomes was a big deal to be able to say, no, this thing is even present in humans, and that makes you realize that it's been conserved to have the same set of 21 letters or 22 letters over almost 1 billion years.
So how significant is that gene? Because it's a big deal. So to say a farmer in Ohio or a housewife in Kansas, how would you explain the significance of that gene in layman terms?
Yeah, so every genome of an animal or a plant has something like 20,000 genes that encode proteins. But there's another few thousand genes that encode these tiny RNAs that are called microRNAs, or other small RNAs that are sort of cousins of microRNAs. And that turned out to be a size regime that nobody had been looking in. And so our research sort of said, no, you need to be looking in that size regime. And it's a little bit like making a telescope that doesn't just look at light, but looks at X-rays, that you start to realize that you can observe things at different wavelengths. And that's been transformative in astronomy. And so we were sort of bringing that concept to biology, looking for different size regimes for genes.
So what are the practical application of that knowledge?
So we've discovered all of these regulatory axes that use these small RNAs, and some of them are mutant in various diseases. And these small RNAs have been used for new kinds of therapies that involve what are called small interfering RNAs, siRNAs. And they're the cousins of the microRNAs. So there's miRNAs and siRNAs that are these very, very tiny RNAs that can sort of almost pass through, they do pass through membranes. And so that it's more easy to use to develop medications based on them too.
Cool. And do people encounter this, the result of your discovery in our regular day lives? Where do we see them?
Yes, in terms of the variation you see in nature, that much of it is due to microRNA regulation. It's the kind of thing that's highly mutable, that will change things like colors of birds and things like that. So but in terms of like medicines that are on the shelf, it's a growing field, but it's not yet the dominant field by any means.
Great. So what inspired you to go on that path? What inspired you to go on the path that led you to your discovery?
The communities of people that communities of scientists that were inspiring to me. But when I went to graduate school, I sort of knew that molecular biology was exploding, and it was right at the moment when the genes could first be purified in a way, and so it was easy to recognize that was a revolution.
And this is a big recognition. How do you see it impacting your career and your work in the future?
The Nobel Prize is a recognition that's sort of 100 times as much press and celebration as any other award. So it's not a part of a continuum. It's a quantum leap.
And there are co-winners. There are two of you who won.
Yeah, Victor Ambros, whom I have yet to be able to find on the phone, and we have been buddies since 1982, when we met at MIT, and we were both postdocs with Bob Horvitz, who won a Nobel Prize in 2000, so that's very sweet. He called earlier today. So for him to have two of his postdocs, and Victor was his first postdoc, and I was either second or third, depending on who came in first that day. And so, that's a big deal for all of us.
I bet. So can you describe the role of each of you in this work, in the discovery? Did you have distinct roles?
Yes, we had distinct roles. So Victor did much of the genetics on this gene called Lin4, which is the first microRNA. And so he figured out that Lin4 encodes a very small RNA. And I figured out that the target of Lin4 is another gene that it can base pair with. And Victor and I figured out that these two genes are sort of complementary. They kind of kiss each other on the, and that's how the process works. And so that was the first microRNA and target that came together. And then 7 years later, we got a mutation in the second microRNA, not looking for it to be a microRNA, and that one was conserved all the way to humans. And so that's when it became clear that it was going to be general and not just something peculiar to this little worm.
Great. And how does it feel after all those years to see the global acknowledgement of the groundbreaking nature of your discovery?
Yeah, it's wonderful to get that kind of recognition, and surrealistic to get that kind of recognition.
So were you awake this morning when the call came in?
No.
Tell me more about the moment.
The phone. Well, when a phone rings at 4:30 in the morning, it never happens here. So we answered.
And what did they say? What was your response?
It was, I'm the secretary of the Nobel Committee, and I bear good news. And I sort of knew when I answered. And Natasha, she figured, my wife figured out that it was, she goes, it's a Swedish accent.
So, I mean, you guys, I believe you guys suspected that you might have been in the shortlist, right?
Oh, sure, yeah. When you win other prizes, you sort of figure that there's a chance.
He won the Predictors. There's like a whole series of Predictors, and he won all the Predictors.
Yeah, but you know, not everybody. People can be over. They can pass over you, and the Nobel Committee has its own tastes of what they like and don't like.
What did they say they liked about your work?
Well, they don't say, well, I wish you'd done X, Y, and Z instead. But no, they're very effusive about the thing, and we've been to 2 other Nobel Prizes. Right, we went to Stockholm to see Bob Horvitz get his, and that was just wonderful to see your teacher be anointed for his work. And then we went when our buddy Jack Shostak got the Nobel Prize, and so we've been twice, which is twice more than I would have expected to be in Stockholm. No, you don't just show up to the Nobel.
So now there's a 3rd trip coming up, right?
There's a trip coming up. It'll be the 3rd, possibly the best.
I bet. And there's a cash award, right?
Yeah, I don't know the details of that, but I'm sure there is money that comes with it. Yes, but the money is great, but the notoriety is what's very special.
The honor.
I bet it's priceless.
Yes.
So what? You've had a few hours to process this announcement. So, what's on your mind now that you've had time to really digest the news and the reality to sink in?
Well, it's a pivot in your life, right? So, there's a lot of public outreach with it, right? There's a lot of talking to people about science.
Anything else we should know?
No.
Yeah. Cool. Really appreciate it. Thank you very much.
Okay.